volunteered to tell you a little bit about SOD models. And everybody uses the G93A model, but there are other possibilities out there. Oops. All right, so SOD1, this is a schematic diagram of the structure. There's more than now 170 mutations in this enzyme can cause ALS. So G93A is just one of 170 choices you might have to make a mouse that would have ALS. We've looked at about 40 different variants, and all of them that we've looked at, the mutant protein is more prone to aggregate, and it will make a pathological inclusion structure, at least in cultured cells anyway. And all the mice that have been made also have SOD1 positive inclusions. So SOD1 normally is a homodimer that binds copper and zinc. The wild type enzyme is very, very stable, very well folded. It's expressed everywhere throughout the body. All right, so one of the main things about SOD1 ALS that I find very interesting is that you can predict how fast a person is going to progress in the disease by what mutation they have. So people who have, in North America, the most common mutation is the alanine 4 valine mutation, alanine position 4 to a valine. People who inherit this mutation from the onset of their first weakness until the time that they are respiratory failure is average 1.2 years, plus or minus 0.9, which means there are some people that go down extraordinarily fast, a few months. Others who inherit G37R, for example, or H46R, their progression is much, much slower. They decline much more slowly. They don't reach respiratory failure until for 17 or 18 years. So between those two spectrums, there are you have a whole range, but those are the relatively extremes. And this is quite a bit different from, many other degenerative diseases where mutations in the gene cause the disease. It's the age of onset that is modulated by the mutation. So the more severe mutation has an earlier age of onset. In this case, the age of onset across all these individuals is not much different by mutation. They all average out to around 52 or so, but it's the duration which is different. So how fast they progress, which is different. All right, so SOD1 mice. The G93A mutation was the first one that was put into a mouse by Mark Kearney. And for that construct, that mouse, they used a piece of human genomic DNA, it's 12 kb. It has all the exons, introns of the human gene, and mutated one site, exon 2, to make the lysine arginine mutation, and that mouse got ALS. It was a science paper in 1993. And this mouse, because it was the first, is the one everybody's used. It just so happened this mouse is a very robust model. It had many copies of the SOD1 transgene, and the mice express probably 10 times the normal level of SOD, and they go down in about 4 months.
Can I ask a question on that?
Yeah.
If you look at the prevalence of these mutations in the population, A4V, is that the most prevalent? And what's the prevalence of G93A?
So A4V is the most prevalent in North America. There's been about 84 documented cases of A4V. G93A is far less prevalent. It's mostly in Europe. It's about 11 people. Last number I knew of was 11 cases. So, all the ones I've listed here are examples where a mouse has been made. G93A was the first, and there's G37R, G85R by Don Cleveland. There's an A4V mouse out there that Tikra Sadiq and Hanning Zhang made, but it didn't express very highly. It doesn't get any symptoms on its own. There's an LA4V mouse. There's several mice that have mutations that replicate truncation mutations in SOD that cause ALS. So these produce truncated products. These mutations all end around exon 5. And for all of these animals, the age of onset of the mouse is fairly proportional to how much transgene is expressed. G93A are still among the earliest. All the others, the age of onset to paralysis, or age to paralysis, ranges from around 6 to 8 months out to around 14 months, depending on which line you use. The one very what's somewhat disappointing thing about the mice is the mice don't model the variations in disease duration that people have. So the G37R mouse that was created by Phil Wong, the interval between onset and paralysis is not much different from G93A. So G93A is a mutation associated with fast progressing disease. G37R is slow, but the mice don't model that duration aspect at all, as far as anybody can tell. H46R has been produced in both mice and rats, and in both cases the duration is not much different from G93A animals. So mice, the level of expression dictates onset, and then the duration is fairly proportional to the onset. So G93A mice, which go down very fast, have maybe a 3 or 4 week duration. A mouse like 85R that gets paralyzed at 14 months might have a 8-week duration. It's because everything is delayed and slower. All right, so one of the even though all these mice get ALS, there are some some of them have very different properties. Years ago, Jeff Elliott and his group became interested in this protein called copper chaperone for SOD1, called CCS. This protein's job is to transport copper from the membrane transporter, copper transporter, and deliver it to SOD and charge SOD with copper so it becomes active. CCS actually has a domain that's highly homologous to SOD. CCS forms a dimer like SOD. It has this extra domain which is involved in the copper shuttling from the membrane transporter to the enzyme. So Jeff Elliott's group made a transgenic mouse that overexpresses CCS, and the idea was that this could improve the mutant SOD by giving it more copper, which would be a structural stabilizer from the mutant protein. So what they found instead was that they overexpressed CCS, and the line you will look at here, this one right here on this figure, overexpressed CCS, the mice got sick really, really fast. They went down in less than 60 days. And in these mice, what they saw was an exacerbation of a pathology that the G93A mice have, which are these vacuoles, these holes in the spinal cord, which are formed by mitochondria that are swelling. So it's a mitochondrial swelling defect that happens in these, and this is really exacerbated when you add CCS, when you overexpress CCS in the G93A mouse. After some time, Joe Beckman became interested in this model, and it turns out that what's happening, according to his data studies, what's happening is that the extra burden of SOD1, which is it's a very, very strong copper binding molecule, and the addition of CCS, that because copper intake is so regulated, all the excess copper, or a majority of copper, gets shuttled to SOD, and mitochondria complex enzymes requiring copper end up being starved for copper, and that's why the mitochondria are in such distress. And that was became evident partly from studies when Jeff crossed other SOD mice to CCS. So the G37R mouse crosses CCS also gets sick very, very fast. But the GD6R or GD5R mouse, CCS doesn't get sick any faster. And the truncation mutation mouse doesn't get sick any faster with CCS. And the key factor was that this mutant enzyme, G37R, will bind copper very avidly, just like wild type enzyme, but GD6R does not, and the 126 mutant can't bind copper. So the absence of copper binding by these mutants negated the CCS effect. And then Beckman's lab developed this compound called copper ATSM, which is a chelator type molecule, which can deliver copper. And when they treated the G93A CCS mice, the biogenic mice, with copper ATSM, over their whole lifespan, they got basically a rescue of the mouse. No mice got, hardly any mice got ALS. They all died of old age. So basically completely cured the G93A mice by co-expressing CCS and delivering enough copper that mitochondria were happy and enzyme got folded correctly. It's a remarkable outcome. If you don't give the mice the CCS, of course they die very fast. If you give it to them for a little while, they live a little bit longer, but they still die off. And if you can delay treatment a period of time, and you can partial rescue, but complete rescue is given if you have a lifelong treatment, including in utero. So the drug is given to the moms, right, the pregnant females, before they even give birth. Now, if you use copper ATSM on mice that just have the G93A and not this extra CCS, you get a much more modest effect on survival, but you still get an effect. So copper, without the excess, the copper addition is enough to get help stabilize some of the molecules, not all of them, and they still get you get an effect, but it's not as large as when CCS is present. So the thing to keep in mind then about the G93A mice, and this applies also to G37R mice, is that they have two different things going on. You have a copper mitochondrial copper deficiency. In addition to whatever the mutant SOD is doing to drive disease. If you improve folding greatly in this G93A mouse, you can essentially cure it. All right, so I think the point I want to make is that there's more than one SOD mouse available out there. They all have a very similar paralytic phenotype. They all have motor neuron loss. Some of them, but the attributes of the mutant that's expressed can affect the model. And for example, the truncation mutant mouse, we have that in our lab. We made it. We made one of the versions of that. Well, they don't have the mitochondrial problem. It's more of a pure protein folding disorder. And that is, it's very different. It's a different type of model from the G93A. It's a different, mechanistically a little different from G93A SOD. Okay, I'll turn